Swayam Prabha. Digital India. Educated India.
Okay, great. So let's continue the photosynthesis that we were discussing in the last class. So in the last class, we were looking at the the Z scheme of photosystem 2 and photosystem 1 in cyanobacteria and plant chloroplasts. So we saw how the light harvesting system works, and how the light harvesting system contains what are called antenna chlorophylls, and from the light absorbed by them the energy gets transferred to neighboring molecules by a process called exciton transfer, and that whole array like structures we called as the antenna, light harvesting antenna. And then finally, from all such molecules, you have the energy transfer to the reaction center. So we saw the reaction center P680 and P700 in photosystem 2 and photosystem 1. So today what we are going to do is we are going to see one important regulatory step that happens there, and then we will move on to how the electrons that are transferred due to excitation to the electron carriers, like for example ubiquinone getting reduced to ubiquinol, and then finally to cytochrome B6F complex and to the next photosystem. So how the electron lost from the reaction center is replenished from water. So that is our next discussion. So then the third, we will move on to actually synthesizing a carbohydrate molecule by taking the carbon dioxide. So that is the carbon assimilation reaction. So first, let us look at the photosystem 1 and photosystem 2. So photosystem 2, so in this diagram, what you are seeing in this cartoon, so this is the stacked grana-like thylakoid, where these are oppressed against each other, and while this is at the top or this is at the bottom where they are exposed to stroma, also these sides. So if you look at the diagram, you don't see this photosystem 1 in the oppressed region. Okay, so here you don't see it. And you see photosystem 2 primarily only in that region. And you have this light harvesting antenna that is harvesting the light energy for photosystem 2 are in close proximity. So this regulation is required because the energy required, like this one, photosystem 1 actually gets excited at wavelength 700, requiring lesser energy than the one that required by the photosystem 2, which is 680. So as a result, the exciton harvesting by this light harvesting complex can actually be lost to photosystem 1 more readily to photosystem 2. So these light harvesting antenna could actually be transferring the exciton energy into this one, because this requires less energy than this, because this is excited by wavelength having less energy, P700. Okay. So as a result, what would happen if they are randomly uniformly distributed, then photosystem 2 will not get excited frequently. So this will be under-excited chronically, while this will be over-excited. So if this is over-excited, then the electrons lost from this will not be fulfilled, because the electrons lost from this comes from plastocyanin, which ultimately gets it from the excited photosystem 2. So to avoid this problem, there is a regulatory mechanism that plays a role, and that is what is shown by this asymmetric distribution. So the photosystem 2 is present tightly in a stacked regions of the thylakoid membranes in this granola-like structure. And this tight oppressing of these membranes is mediated by the light harvesting antenna of photosystem 2 itself. And as a result, these are immobilized, and this ensures the spatial suppression from photosystem 1 ensures that the exciton transfer from LHC2 actually goes to photosystem 2. This ensures that the photosystem 2 is adequately excited. So when you have bright light with the heavy component of the blue region, then you have this excited more frequently than this, and therefore the ubiquinone is reduced to ubiquinol form, and reduced ubiquinol form is very readily available. And the electron transfer from this to the next step is therefore needed to be speeded up, because you have more UBH2, and from that the electron should flow via the cytochrome B6F to blastocyanin and to photosystem 1. And if the UBH2, that is a fully reduced ubiquinone, if its level is very high, meaning this system is getting adequate excitation, then that activates an enzyme as shown in this cartoon, a protein kinase that phosphorylates a threonine residue on the LHC2, and that leads to a conformational change such that it is no longer anchored on the other membrane of the thylakoid, and it detaches. And when it is detached and this structure is relieved, then this is free to go and harvest photons for photosystem 1. Okay, so now the UBH2, fully reduced ubiquinone produced by photosystem 2, gets consumed by this. On the other hand, when you have dim light with more component coming from the red region, then this will deplete of the reduced ubiquinone, and then you will have more oxidized ubiquinone, and the presence of more oxidized ubiquinone drives the opposite reaction by activating this enzyme protein phosphatase. And when the threonine is dephosphorylated, then that leads to a conformation change in LHC2 such that it anchors to the adjacent membrane and it gets tightly oppressed. Then it is locked in position with the photosystem 2, and whatever little light that it gets, the energy absorbed is ultimately used to activating or exciting photosystem 2. So this way the light, depending on the light intensity, the light harvesting is regulated such that you have a balanced or optimum production and consumption of the ubiquinone. Essentially not consumption of ubiquinone, the oxidation reduction of ubiquinone to ubiquinol and back. So that is how equalization of electron flow in photosystem 1 and photosystem 2 is modulated by light itself. So light ultimately activates these two enzymes. Bright light activates this, dim light activates this. So this is what they call as larceny, meaning getting siphoned off, or photosystem stealing the electrons from photosystem 1, stealing it from photosystem 2. So that's where they use the term larceny. So that is about the regulation of how electron flow in photosystem 1 and 2 happens. So next let us look at the cytochrome B6F complex itself, which is equivalent to the mitochondrial complex 3. So this is again, we have already seen this. So we saw this with the purple bacteria, which has only photosystem 2. And then we saw green sulfur bacteria, which has only photosystem 1. So there again, we saw the electrons from the excited state flow via the cytochrome B6F complex back to the reaction center. So, and this structure, I told you at that time, is very similar to the one in mitochondria complex 3, which oxidizes the fully reduced ubiquinol, essentially it's called ubiquinol, by transferring electrons to cytochrome F on the mitochondrial inner membrane side. Okay, so where then the cytochrome C moves to the complex 4 and transfers the electron there to oxygen there. So here a very similar structure. So the first part on the left side, you see without the amino acids, without the protein backbone, we see only the prosthetic groups. And you have the heme moieties here. So the plastocyanin is the final one. This is a protein. Okay, so although the name kind of resembles to the pigments we learned, but this is a protein which is functioning like cytochrome C of the complex 3 in mitochondria. So electrons finally move to this plastocyanin, and plastocyanin transfers to photosystem 1. So that has, so before that you have this cytochrome F with this heme F moiety. Then the cytochrome B6 itself contains a heme B and an iron sulfur center. Then you have the heme BH and then another heme called heme X. And there is also a beta carotene, although we currently do not understand the functional contribution of beta carotene here. And the electron flow diagram is shown by this blue arrow. So this is the reduced form of ubiquinone called ubiquinol, and from there through a Q cycle mechanism that we'll see once more in the next slide, but we have elaborately discussed that with respect to complex 3 in mitochondria. So from there electron flows this way and you get the oxidized ubiquinone, and then finally to cytochrome this F, and from that to blastocyanin. So this is the path of the electrons here, and during this process the conformational changes that happen in this complex due to the electron flow ends up pumping protons across the membrane, just like what complex 3 in mitochondria did. And on the right side you have the structure having the protein. So it is a dimeric structure that you see here. So this is the this portion on the top on the luminal side or the P side, the positive side, you are seeing the cytochrome F with the heme F that's shown here. So this one, this structure here. Okay, and the other molecule, this is a dimer, so the other heme F is here, this one. And the other hemes, these heme B, heme X are shown here, and so it forms a canal-like structure or a cavern or a valley-like structure, and that allows the free diffusion of the ubiquinone or ubiquinol in this structure. So this is the Q cycle, the very same thing that we saw in a very abbreviated way. There we saw two separate UBH, reduced QH2's binding, and how one electron goes to cytochrome C and the other one gets cycled back to reduce the semi-ubiquinone that was formed due to one electron transfer. So the same thing here again, from the fully reduced one you have one electron going via this iron sulfur, again this rice iron sulfur protein, to cytochrome F to plastocyanin. Okay, so this is a copper containing enzyme, but it is not similar to complex 4 in mitochondria. And the other electron via this cytochrome B6 through these two heme moieties go back to reducing that free radical version, the radical version of the partially reduced ubiquinone called semiquinone. And that whole detail is not shown because we have already seen elaborately. And a similar Q cycle operates here to ensure 2 electron donation by the fully reduced ubiquinol, but transferring only 1 electron at a time to plastocyanin. In this case, this space, so this area is extremely narrow. Okay, so compared to the stromal side, when I showed you the chloroplast structure with stacked thylakoid membrane, green structures inside orange that you saw, and inside that green is what this space, this lumen. This is really small place, and as a result, when the protons are pumped into this by this complex, the pH gradient really is about 1000-fold. Okay, the proton concentration inside the thylakoid lumen is so high. It's pH 5, and outside in the stroma it is pH 8. And therefore there is a 1000-fold difference in proton concentration, sufficient enough for ATP synthesis. Okay. So our next main topic in photosynthesis, the light reactions part, the first part of photosynthesis, is this water splitting by the oxygen evolving complex or water splitting complex. So it's known by both the names. So essentially the primary thing here, what happens is 4 electrons and 4 protons are taken away from 2 molecules of water. Okay. And the 4 protons are pumped into the lumen. So this is the second event of proton pumping that we are seeing. One, when the excited electron returns, rather moves to the photosystem, one via the chain of electron carriers. In that pathway, we just saw the B6F cytochrome pumps protons. And the next one that we are seeing is the water splitting complex itself. And the 4 electrons, so 4 protons are pumped into the lumen, and the 4 electrons that are taken up, they go into reducing this manganese ions. So manganese can exist in a stable oxidation state between +2 and +7. So it can very readily take 4 electrons in one go, or it can take 1 electron at a time, readily 4 electrons in sequence. And it can also get oxidized by giving out 1 electron at a time or giving 4 electrons at a time. And that flexibility of manganese ion is very smartly co-opted by this water splitting complex. Okay. And that is how the 4 electrons that are taken away from 2 water molecules are given 1 electron at a time to the reaction center. If you remember the cartoon that we saw in the last class or the class before, that green vertical rectangle like things, when an exciton energy is transferred, like 1 photon equivalent is transferred, 1 electron is donated by the reaction center, creating the electron hole there. And that's how the charge separation happens. So it loses only 1 electron at a time, and from the electron donor it takes only 1 electron at a time. But here when you split water into oxygen and protons, you get 4 electrons in one go from 2 molecules. When you produce molecular oxygen, you end up consuming 2 molecules of water. And these 4 electrons, the reaction center cannot take in one go. It will take only one at a time. And that situation is what is very ingeniously handled by the water splitting complex, taking the multiple stable oxidation states of manganese ion into good advantage. And the electrons from the manganese ion is transferred to tyrosine residues on this complex. And so the textbook says the crystal structure solving of this complex was one of the most challenging among crystallizing and solving structures of proteins. And the mechanism has not been fully deduced. Okay, so this is the model that is supported by the available experiments, and this is what currently we know. But there are still some nitty-gritty details that needs to be filled. And this tyrosine residue is the one that takes up the electrons, and from the tyrosine residue, it goes to the next, the reaction center, that is P680. Okay. And this is how from water electrons are transferred into the reaction center. And from the reaction center, of course, it goes via the carriers and finally reducing NADP into NADPH plus H plus. So hydride transfer, like 2 electron, 1 proton transfer happens at the end of the 2 photosystems. And so that is where the electrons have gone. So they have gone, and the protons have also been taken up when it comes through the ATP synthase. Then you have the reduced equivalents, NADPH, generated there. And when the protons are returning, ATP is also produced. So you have reducing equivalents for the dehydrogenase reactions, and then you have the energy to drive them, ATP, both are available. So that it can now readily go and do carbohydrate synthesis. Like, in loosely speaking, opposite of what we have learned so far through glycolysis and TCA cycle. I am saying loosely because it's not the exact reversal. And in the process, from water you have the oxygen evolving out. And this is how the atmosphere got its oxygen. From water only you get it. Okay, it is not from carbon dioxide. And similarly, we saw in the mitochondria, the electrons coming from the donors like NADH or FADH2, succinate dehydrogenase reaction, they finally go to reduce oxygen to water. Okay, the exact opposite ends up happening in mitochondria. And there carbon dioxide is liberated from glucose kind of molecules. Here the carbon dioxide, now we are going to see, is going to be used to make glucose kind of molecules. So we will not discuss the ATP synthase in great detail, but because it's essentially the same as what we saw in mitochondria. You have the FO subunit, oligomycin sensitive subunit. So do not call F0 or F0. And you have this F1 ATPase and the same rotational catalysis. Instead of inner membrane space of mitochondria, intermembrane space of mitochondria, here you have the thylakoid. And instead of matrix of the mitochondria, here you have the stroma of the chloroplast. Okay, this open, this bluish area. So this cartoon actually is a summary of whatever we have learned. So you have the light reaction, here light exciting and all the antenna, all that we know here. And from here, you have that B6F pumping proton into this, and this is the water splitting complex itself, producing protons and evolving oxygen. Then you have it flow into B6F, and from that to blastocyanin, and blastocyanin is exactly like the cytochrome C in mitochondria. This is again on the surface inside. There that is on the intermembrane side, here it is on the luminal side of the thylakoid. to photosystem 1, which again get excited by light as well. And from there, the electrons go via ferredoxin to ferredoxin oxidoreductase reduces NADP to NADPH. So that is where from water the electrons have gone into. So from here, this will be used and combined with the oxygen, the carbon dioxide to make the carbohydrates. And the proton gradient created here by these two water splitting complex here, and then this cytochrome B6F complex, that when it flows down the gradient, then you make the ATP. So reducing equivalents as well as ATP are produced by the light reactions. So up to now, the light is absolutely essential for these reactions to happen. Essentially, we have light energy now in chemical potential energy. Okay. So this is like the guy standing on the tall diving board. Okay. So potential energy is very high. And the required electrons and protons are here. Okay, a small detour into evolutionary history before we go to the carbon assimilation reactions. That is a simpler a pump, proton pump, which is directly activated by light. Okay, so there is no such complex thing like this, where you have a separation of activation and pumping, etc. So this one, what it does is, this molecule is called bacteriorhodopsin. Rhodopsin simply because, like the protein in our rods and rod cells in the eye, in the retina. So we already know retinal, retinoic acid, retinaldehyde, all that, because we have learned that in the studying the fat soluble vitamins. So there I told you the cis-trans isomerism happening in the double bond of the retinal, changing from cis to trans activated by light, and then that returning back to cis. That alternating the cis-trans isomerism of retinal is used for sensing light in our eye. A very similar thing here, and that is why this is also called a rhodopsin. Opsin, optical, so it is about the light, and then this rod cell is probably from where this rhodopsin name comes. So here, very similar thing. Only thing is here there is no brain sensing and making an image. Instead, this cis-trans isomerism happening due to light absorption by this retinal is used to alter the conformation of this protein such that the amino acid side chains end up donating, accepting protons, and eventually leading to proton pumping. Okay, so the light energy alternate the cis-trans configuration of the retinal, and that changes the conformation of the protein such that the protein ends up pumping proton. Okay, and this is helpful for bacteria living in extreme saline environment. Okay, so the extremely saline environments can have salt concentration as high as 10 power 4 molar or 10 power 5 molar sodium chloride. In such a high salt concentration environment, although this bacteria like us uses oxygen and burns fuel like glucose available from the surrounding media to obtain its ATP and energy requirement, the oxygen availability in such environments is very low. And due to that, it supplements its energy requirements by using this protein and making some ATP synthesis. So when light directly activates bacteria rhodopsin and pumps proton, then you have a proton gradient generated. And when that proton gradient goes via the ATP synthesis, you get some ATP produced. Okay. No reducing equivalent, no other carbohydrate synthesis, nothing. A single molecule responding to light and undergoing conformation change pumps proton across, and the proton gradient is used to make ATP. So this is how it supplements the normal oxidative phosphorylation by a means without requiring the oxygen. So under low oxygen condition, this bacteria benefits by this supplemental ATP synthesis. Okay, so halophilic means salt-loving bacteria. So these are so adapted to the salty conditions, they cannot live in less salt solution, leave alone fresh water. So there are places on Earth where you have all sides surrounded by mountains. So therefore, whatever little rain that falls on the mountains, they come to the middle bowl-like valley, and they no water flowing out from there. It never rains to fill the whole bowl and overflow, and the water evaporates there itself and leaving the salt behind. Okay, so the rain falls on the mountains, dissolves the minerals and salts, comes to the valley, forms a lake, and when it is summer, the water evaporates, but the salts stay there. And in such environments, the whatever water that remains on that salt, they are called a brine, B-R-I-N-E, brine. That's extremely salty water, and in such environments you find this bacteria. So a little bit detail about this proton pumping. This is primarily because we learned what is proton hopping, right? This water is slightly ionized, and that proton is readily taken up by a water molecule forming a hydronium ion. And since water molecules are hydrogen bonded, such protons resulting from ionization through this hydrogen bonding can readily hop instantaneously to long distances. And that time I told that kind of proton hopping is really very useful for catalysis at the enzyme active site. And here is one such example we see here. So in this protein, so here this is the retinal molecule, and in the dark condition, this lysine side chain, this protonated Schiff's base this has a high pKa that is ensured by the protein conformation. And upon illumination, the retinal structure changes such that the amino acid is positioned closer to another one, and that conformation change leads to reducing the pKa of this group Schiff's base nitrogen, and that readily donates the proton because its pKa value is reduced, and therefore even at a lower pH it readily donates proton. And that via a series of proton hopping shown here, from threonine 89 to aspartic acid 85 and 82 and so on, it comes to the other end of the membrane. So this is the membrane. And finally, when these things undergo conformational change, their pKa also changes such that they donate the proton. Only thing is, when they donate the proton, it is into the cytoplasmic side of the bacteria. Okay. I'm sorry. Through the extracellular side, in the periplasmic phase. So that is where it ends up pumping. So between the cell wall and the cell membrane, you create a higher concentration of proton, and compared to the cytoplasm. And when it returns via the ATP synthesis, that leads to energy synthesis, ATP synthesis. So So the main point here is the conformation change affects the pKa of this group as well as this. And as a result, upon illumination, the lowering pH leads to proton donation, and that via these amino acid side chain enabling proton hopping, it comes to the other side. And since this one's pKa also is reduced, it readily donates the proton, but that proton is donated into the periplasmic space. Now this proton donated by this needs to be fulfilled, and that is done by another amino acid, in this case aspartic acid 85, and that eventually takes that proton from the cytoplasmic site. So essentially you are pumping from cytoplasm to the periplasmic membrane protons whenever illumination happens. So this is how the bacteriorhodopsin works. So well before going and studying the photosystem 1, photosystem 2, etc., people focused on the bacteriorhodopsin because it's an extremely simpler protein and easy to purify, crystallize, like relatively compared to the other ones. And so this is one of the light sensitive molecular machines that you encounter. There is one more, photolyase, that helps in thymidine dimer repair in DNA, so that you probably will learn in molecular biology. Okay, so that is all about light reactions. So now we are going to use the reducing equivalents produced in the form of NADPH and the chemical potential energy in the form of ATP, and we are going to hydrolyze the ATP, and the energy released will be used to drive the reduction of carbon dioxide into triose phosphates and then into hexose phosphate, etc. So that is what is going to happen in the carbon assimilation reaction. And for this, you do not need light. It can happen in the presence as well as absence of light. So this cartoon gives you an overall view of what is happening. Okay, from carbon dioxide and water, you have the triose phosphates formed, and from them to hexose phosphates. And this may be converted into disaccharide sucrose, and that is transported to other parts of the plants, like to root and storing in the roots. And sometimes in the stem itself, like for example, sugarcane. That's why sugarcane juice is sweet, because sucrose is there. So sucrose is the form in which it is transported. So that's an important concept you need to remember. So the form in which the carbohydrates produced in the leaves are transported to other parts of the plants is in the form of sucrose. And in grains like rice and wheat, barley, etc., or potato, cassava, in all of them, the storage is starch. Glucose, and when we know that starch is a polymer of glucose. And for cell wall structure, you have cellulose. And this one pathway that we didn't cover during our carbohydrate metabolism, we saw glycolysis and we saw TCA cycle, but there is one more pathway called the pentose phosphate pathway, or PPP, triple P pathway, or HMP shunt, in honor of the scientist who discovered that pathway. So essentially that pathway, we are not going to go into it. I am briefly telling you what is the essential function of that pathway. That pathway produces pentose sugars from the hexose sugars. And the pentose sugars we know are required for nucleic acid production. Okay, and the other intermediates for protein as well as lipid biosynthesis. So some of the hexoses can go into this. So when you look at all of this, you see these molecules, this and this. So entire set of macromolecules are covered. Molecules of like whatever we learned are all covered. So meaning all of them are produced from carbon dioxide fixation into triose. So this is why carbohydrate chemistry is central in life. Okay, so life is actually carbohydrate chemistry. The other molecules are all produced from carbohydrates only. That is because the way carbon enters into the system is via carbohydrate formation. So now let us look at this. We are not going to learn all of this. Okay, so our focus will only be coming up to this. So we are going to assume, oh yeah, this is self-explanatory. We'll understand, and also this is the only biochemistry course I'm going to learn. So I'm not going to cover all the branches and details. Okay, before we go into this, again, what is the stage in which this drama happens? So that's what we are going to first look at it. So chloroplast is where all this light reactions, all the green thing. So this is where NADP, its production, ATP, all that is happening, right? And they are used in the stroma area here, and that is where the carbon assimilation reaction related enzymes are located. And at the end of this, when you produce a lot of carbohydrates, they are converted into starch, and that leads to conversion of this chloroplast into what are called amyloplasts. Amyloplasts do not have this internal membrane structures. These are all broken down, and the entire space is used for storing starch, and such starch filled structures, these organelles are called plastids. Okay, chloroplast is a plastid. Here in the title you have the spelling. So the plastid filled with the starch and not doing photosynthesis is amyloplast. So here is a micrograph of amyloplast. Iodine stained, iodine binds to starch, and you have here the starch granules shown everywhere here. So this is starch filled. So this is the kind of plastid you will find, for example, in potato. Okay, potato is not going to have this green chloroplast. And these actually exist in homeostasis. Which form this exists reflects what tissue and what what organ and what its function is. For example, in leaf, where photosynthesis happens, they exist in the form of chloroplast. In storage organs, they exist in the form of amyloplast, meaning these are reversible. These structures, an intermediate called proplastid, can assemble the grana and they can become chloroplasts. Okay, and chloroplasts can lose them and become protoplastids, and from there they can go and become amyloplasts. So this is an intermediate structure called pregranal plastids, meaning granum has not formed, but the membranal discs, flattened discs have already formed. All right, so this is the overall summary of the carbon assimilation reaction, like the way we saw the TCA cycle circle first, and then we went into the individual reactions. Similarly, this is the overall perspective. So the scientists who worked out this, there were actually three of them, Belvin, Boschem, and Calvin. And in that three-member group, Calvin is the main person. So he got the Nobel Prize in 1961 because this is one of the central aspect of biology. Okay, so this is how carbon enters into the biosphere. So carbon dioxide, in sense, should be called inorganic carbon because it's not part of the living system. So, but carbon in this form, like these carbohydrates, they are genuinely products of life, and therefore they only are organic compounds, organism derived. So this inorganic carbon becomes organic carbon by this cycle. So this cycle has three stages. The first stage is carbon fixation. The carbon dioxide gets incorporated into a pre-existing carbohydrate, and this pre-existing carbohydrate used here is this pentose or ribulose 1,5 bisphosphate. Ribulose, okay, that UL should sound aldolose like fructose, okay, it has the like fructose, it has a ketone group. So this will form hemiketal if it forms an internal a linkage. So ribulose is phosphorylated in 2 places, so it's ribulose bisphosphate. So this 5-carbon compound becomes 2 3-carbon compounds by taking this carbon dioxide. So this is the fixation reaction. And next there is a reduction reaction because the end product is 3-phosphoglycerate. This we saw in the reverse order in glycolysis. Glyceraldehyde 3-phosphate became that is 1,3-bisphosphoglycerate. That's what it forms. So that phosphoglycerate is what we are seeing here. In glycolysis, it was the opposite direction. So now it is in this way. So this is a reduction reaction because carboxylic group, aldehyde group. So that's a reduction, carboxylic acid to aldehyde group. In glycolysis, we saw aldehyde becoming carboxylic acid and therefore we called it as oxidation. So that's a dehydrogenation reaction as well there. So here it is the opposite of that. So therefore we call this reduction. So that's the second step. Third is regeneration of the ribulose bisphosphate. Therefore the cycle can continue. And to take care of the stoichiometry, we begin this way. We begin with 3 molecules of this. So 3 times 5, 15 carbons. And we take 3 molecules of carbon dioxide. So 18 carbons end up producing 6 3-carbon phosphoglycerates. Now of the 6, 5 of them are going to combine. So 5 times 3, 15. So 5 carbon, 3 molecules. Okay. The net result is 3 carbon have become 1 glyceraldehyde 3-phosphate, which has 3 carbons in it. So this is the stoichiometry. 3 carbon dioxide are fixed with 3 ribulose bisphosphate, producing 6 trioses. Okay, whether you are talking about this or this, 6 of them, of which 5 are going to make 3 of this pentose, and 1 is the net gain. And that 1 is going to now form hexose and go into making starch and sugar, etc. So we have the fixation step, reduction step, and regeneration of the acceptor step. That regeneration step requires the energy in the form of ATP. So I'll stop here. In tomorrow's class, we will focus on the fixation reaction. How ribulose 1,5-bisphosphate is converted into 3-phosphoglycerate.